So it's my pleasure today to introduce Dr. Ruth McPherson. She received her PhD from the University of London in the UK and MD and subspecialty training in internal medicine and endocrinology at the University of Toronto. She has held academic positions at the University of Toronto, McGill University, before joining the University of Ottawa Heart Institute. She has held continuous funding during her career from agencies including the NIH, the Heart and Stroke Foundation, and CIHR. She has published over 270 manuscripts in peer-reviewed journals, including quite a number that are in very high impact journals. Dr. McPherson's laboratory research is centered on understanding the genetics and genomics of two complex phenotypes, obesity and coronary artery disease. Her research has been recognized where she has received multiple awards, including one from the International Atherosclerosis Symposium and an Outstanding Achievement Award, an AHA Council Special Recognition Award, the George Lyman Duff Memorial Lectureship, the UBC Margulies National Heart Disorders Prize, and the Canadian Cardiovascular Research Achievement Award, just to name a select few. She was elected to fellowship in the Royal Society of Canada in 2014. Dr. McPherson directs the Lipid Clinic, which was really a visionary approach when she did this, a model to be embedded with cardiologists. And this model is now being implemented across the country, where endocrinologists and cardiologists work hand in hand together to manage patient care. She also manages the Atherogenomics laboratory and the Ruddy Cardiovascular Genetics Center. I can tell you she's in an elite class of scientists who can seamlessly synergize findings from the genetic, biochemical, clinical, and population levels. Her group has contributed significantly to identifying loci involved in cardiovascular disease. And as one example of the continued impact of identifying these novel genetic variants, her team has put considerable energy into understanding PCSK9 variants in hypercholesterolemia. And these now may have far-reaching implications for cancer, with the discovery that PCSK9 may also modulate MHC class II receptors. More recently, she has also collaborated with Mary Ellen Harper and Bob Dent to understand the genetics of diet-resistant obesity. And we are beginning to see some of the high impact fruits from that collaboration. So it's really my pleasure today to welcome Ruth to weekly research rounds and listen to her talk entitled Coronary Artery Disease, From Genetics to Drug Development. Welcome, Ruth.
Thank you for that very kind introduction. I mean, that just tells everybody that I'm very old and I'm going to retire in like 3 years. So there you go. Okay, so these rounds are always a bit of a challenge because I'm trying to kind of make them comprehensible to more than laboratory-based scientists or clinicians. So this is a little bit of a overview of what we do and what I think is important in terms of what we've learned with our genetic studies over the course of the last many years, actually. Oh, doesn't mind do that. There's another way to do it, I'm sure. Okay, how do you advance? Oh, is it a touch? How do I advance the slides? Doesn't want to do anything. Oh, I have to. Okay. Okay, just so. Okay, perfect. So I'm really going to talk about two kinds of genetic studies, and our laboratory has been highly involved with Alex in the audience in genome-wide association studies. And these studies have given us, a lot of new information because we're looking at the cumulative effects of multiple common variants. So particularly important in terms of understanding new biology or confirming some of the things that we knew. Also of utility in formulating genetic risk scores. So we can now, with the help of the data we obtained from an Affymetrix and imputation into a whole genome data set, such as are available, we can actually get good information on about 15 million SNPs. So we're able to create genetic risk scores for really almost everything we might be interested in. So it's not once you have all of these data on 15,000 SNPs, you can create risk scores for diabetes, for cancer, and for coronary disease. So the other type of genetic studies, and we have been involved in collaborations with other groups, particularly say McPherson in Boston, and that is exome or sometimes whole genome sequencing. So that's a bit different. Instead of looking for these common variants that cumulatively have an effect, the rare variants are, of course, those are most likely to lead us to new drug targets. And that has been true in terms of development of PCSK, really works. PCSK9 as a therapeutic target for lowering LDL. Not a very popular thing. Or, yeah, there we go. Or new drugs such as aminucamab, which is an ANGPTL3 inhibitor that I'll tell you about today, and new therapy for homozygous FH and also for hypertriglyceridemia. So I think any of you who have listened to genetic talks will have seen this slide many times, but it does certainly make the point of what genetics are. So of course we can think about genetic variants as those that have a large effect size. So those are the ones that are likely to be drug targets. And things such as familial hypercholesterolemia due to a mutation in the LDL receptor gene, ApoB, sometimes PCSK9, less often. Those that have a relatively small effect size, but are common. And these are the common variants. So these are the ones we identify by genome association studies. These are the ones we identify by exome sequencing. And then, of course, we have some variants that are kind of in between, sort of moderate effect size and, not rare, but, often the allele frequency is less than 0.01%, 0.01. So this is what we've been doing for many years. And when we started to do this with the help of Alex in the audience, we bought these Affymetrix. And what did they cost? Alex said, you're about like $450 each, something crazy. That's right. Now they're less than 100. So everything we do has become less expensive. So basically we have 1 million SNPs and we can impute from whole genome data sets and we can end up with about 15 million SNPs. And that really covers, most of the common variants across the genome. And there's some things we don't pick up with great ability, such as copy number variants, et cetera, but certainly that gives us a lot of information on the basically polygenic list. So going back to 2007 with Alex and others, we discovered the 9p21 locus for coronary disease, really high effect size and actually relatively common. So that, of course, is the low-hanging fruit. For me, the discovery population were 300 patients who had 3 to 4 vessel bypass at a very early age. So, those are the ones likely to harbor some really constant mutations. So following that, in collaboration with other groups around the world, particularly as part of Cardiogram and now Cardiogram plus C4D, data from the 1000G meta-analysis, UK Biobank data, et cetera, et cetera. And you don't do this without hundreds of thousands of samples and multiple groups around the world. So basically by now we have identified about 213 replicated loci with genome-wide significance. So if you propose that, we're about 1 million comparisons correcting for multiple comparisons. This means that you need a p-value of 10 to the minus 8 for significance. And of course, you know, we're going to miss some things. There are other ways to look at this. If we look at the FDR variants, then of course there are many more. And actually, if you pursue them, many of those FDR variants, less significant, of course, turn out to be costly, but turn out to be indeed real. So really the point to make here in terms of the GWAS identified risk variance is that the population frequency varies a lot, anywhere between 2% to 49%. 49% is 921, but a lot of them are uncommon, but you know, there's some that are in the middle. One problem is the genotype SNP is often not the causative SNP. So it's in linkage to equilibrium with the causative SNP, and that means we have to do all this work in the laboratory to really understand what the causative SNP is. Generally, the effect size is low. So odds ratio 1.03 to 1.25. The only two really common loci with high effect size are 9p21 and lipoprotein A loci. So LPA, lipoprotein A, has been known to be a risk factor for many years, and certainly more attention on LPA as a risk factor recently. So we know the heritability of CAD is about 50%. So about half of your risk for coronary disease is related to the risk factors you know about: smoking, diabetes, hypercholesterolemia, et cetera. But about half of the risk is genetic. So this is why having a family history of premature coronary disease is strongly predictive of your own risk. The other point I should make in terms of these 213 loci is that most of the novel ones are not related to known risk factors. So lipoprotein A is 1921, of course, if not. So this has led to, I think, important new biology. And sometimes it's biology that we knew about. So the variants that, common variants are associated with coronary disease, many of them associate with levels of LDL cholesterol, which is PCSK9, ApoB. All of these came up in the GWAS studies. Triglyceride-rich lipoproteins, so ANGPTL3 and ApoA5 certainly came up in GWAS studies, although the whole genome sequencing was also very helpful. And of course, loci associated with lipoprotein A. So these are really very important. That's not surprising because we have a priori data that is important. What I think is really interesting is that a lot of the new variants associate with, associate with, we're out of practice because we don't do these things anymore. At least I did them on my computer now up here. Associate with vascular remodeling, vascular smooth muscle cell differentiation and migration, and the extracellular matrix. So, you know, this isn't something that we measure in the lab in terms of what's a risk factor for heart disease. So the ones there that I've highlighted, the ones in red that our lab has been working on over many years, and this is a tedious process, I have to tell you, but certainly 9p21, call4A1, call4A2, this NEPA locus, I'll say a word about, SMAD3, fibronectin-1 that Sebastian Sibian in my lab has recently been working on. We just published that in the CBG not long ago. So these all relate to the vascular wall, and I think that has given us new information. I do have to admit that none of these is a drug target because the effects are complex and affect many processes, but it's a process that we're starting to understand is really, really important. Others, of course, relate to inflammation and innate immunity. And not surprising for many of the investigators here at the Heart Institute, certainly the IL-6 receptor, very, very important. Our laboratory has been working on CARS2, and in fact, Anne Tu Dang is not here today because she is presenting that work at the Canadian ASCB meeting in Whistler. So she's back there. But the CARS locus definitely relates to inflammation and really quite an interesting function there. And TRIB1, which relates to triglyceride-rich lipoproteins, also a locus we've done a lot of work on, TRIB1 and the tribal that may or may not control TRIB1, but Sebastian has also been working on that. So many processes. And of course, atherosclerosis is a complex disease. So I'm just going to give you a couple examples. So the thing is, when we get to the lab, most of these loci are not coding. Most of the new variants are not in coding regions. So that, so makes it a lot more difficult to figure out what's going on. But there was one that Tara Mitzman in my lab worked on a few years ago. And this was a coding variant that was actually in this gene, ZC3HC1, that encodes NEPA. And the variant that was discovered, the HIS variant, is actually associated with protection for coronary disease. So really interesting, getting back to cellular proliferation, et cetera. So NEPA is a regulator of cell proliferation. And of course, we now know that abnormal proliferation of arterial smooth muscle cells is really a key feature, not only of atherosclerosis, but also of restenosis after PCI and stenting. So it was interesting because it's coded, made it easy. It had a damaging score when, figure, is this likely to have an effect on the function of the protein? And it's conserved across areas. So this also suggests that indeed it probably is important. So we went on to do, the sorts of studies to do. So this was not particularly complex, but basically she was able to show Tara that the risk variant, the arginine 363, exhibits, first of all, there was less protein expression, greater proliferative capacity, and really putting this risk locus into those that really affect smooth muscle cell proliferation, probably acting as the vessel wall. So the important point there is that although this NEPA variant is one of the GWAS variants we've worked on, there are a bunch of others that also probably a lot of the action is via smooth muscle cell proliferation. The 9p21 locus that we worked on for a long time and discovered some new things, but finally gave up on. But I think Alex is still doing some work on it. MIA3, TRIS1, H81PL1, LMOD1, and SMAD3 that we've worked on. So, really this is a group of variants that probably exerts most of the effect at the vessel wall and related to smooth muscle cell proliferation. So that was fairly easy to work on. Most of the variants we've discovered are actually in non-coding regions. So that's not nearly as simple. So first of all, you have a novel locus. So the GWAS, they come up with a SNP. That SNP is not necessarily the cause of the SNP. It may be a linked-specific equilibrium with a SNP that actually has a cost effect affecting gene expression, transcription factor binding, something else. So there are all of these databases available that those in the lab we know all about. Genome, UCSD genome browser, and code data, lots and lots of public databases are useful. And, going from these to functional studies is really where all of the laboratory work is. So I always tell everyone that the GWAS studies, that was the easy part. The difficult part, of course, is what we do in the lab, and that takes a long time. So I'll give you an example of Adam Turner, who's now at the Gladstone within my lab, but he did his PhD with me. And so he decided to work on the signal in SMAD3. And so what's interesting here, it's a intergenic locus. So, and actually the lead SNP is here, but the lead SNP clearly in linkage equilibrium with other SNPs. So basically what Adam was able to show is that it was that in linkage equilibrium with SNP called the RS152 SNP, and he was able to show that this SNP is expressing QTL. So if you can find out that the genetic variant affects the expression of an adjacent gene, it certainly suggests, it doesn't prove, that that is likely to have a causative effect. So we went on to do a bunch of studies showing that the protective allele of this SNP really disrupt an AP1 binding site in a SMAD3 intron enhancer and reduced enhancer activity. So that all sounds very easy, but it took a long time to do that. We then went on to show that inhibition of AP1 activity upstream to show that the allele-specific cancer actually requires AP1. And then CHIP experiments to show preferential binding of a bunch of AP1 proteins to the C allele. So he then showed that the protective allele reduces SMAD3 expression and alters the muscle cell proliferation. So it's really just a kind of a little story of how you go from a SNP that is pulled out of a GWAS study to finding what's likely to be the causative SNP based on functionality, and then doing the studies to actually show how it works. So, that study takes 2 years, whereas a GWAS study didn't take that long. Okay, so beyond that, beyond simply what we've done in the past, looking, variants that are more frequent, associated significantly with coronary disease or some other phenotype that is of interest, there are a bunch of computational approaches. I won't talk about all of them today, but one is pathway analysis, and then there are a lot of multi-omic analysis that can be done. A lot of that being done by Majid Nikpay in bioinformatics. So pathway analysis is kind of interesting, and I think it's really, really important in terms of biology. So, of course, perturbations with these complex molecular networks and metabolic cellular pathways, and these are likely to be involved in both the initiation and progression of coronary disease, other complex phenotypes. So I told you that a variant is only significant if p-value of 10 to the minus 8, but less significantly associated variants, really can provide important information regarding biology when those variants cluster within a pathway. So, small differences can be important if it's going to affect the function of the overall pathway. So we did this a few years ago, and basically you have to do these things with, stage 1 study and pulling out p-values and then replicating another study. So the first set, we had 7 studies and, a total of about 20,000 individuals. And we selected the nominally significant pathway. So p-value 10 to the minus 5, FDR less than 25, so fairly liberal. And then did replication in a much bigger data set and did GSE analysis on stage 2. So we come up with replicated pathways. So a p-value less than 0.01. So once again, we're doing pathways, we're not doing SNPs. And an FDR less than 5%. So this information, it's really consistent with our other approaches. The pathways that turn out to be most significant in that analysis are, lipoprotein metabolism, that if it's not there, you're kind of concerned. But axon guidance pathways, which are really interesting, 7A. So basically pathways that can affect movement. Axon guidance, we think of it mainly in the neuronal system, but certainly also at the vessel wall in terms of efferocytosis, etc. Once again, extracellular matrix comes up, innate immunity, muscle cell proliferation, and TGF-beta signaling. So I think this is interesting. It is certainly complementary to the other kinds of studies that we've done. So out of those studies, these are the things that really come up: innate immunity, macrophage inflammation, TGF-beta signaling, smooth muscle cell proliferation, lipid transport, and the integrity of the extracellular matrix. So I think, this is really very important in terms of atherosclerosis, because, I see patients all the time. Interesting, see a patient who's never had an LDL greater than 2.2, who has never smoked, is not diabetic, but, has significant three-vessel disease. So, what makes that individual more susceptible to what is really a modicum of risk factors? And obviously the integrity of the vessel wall, the extracellular matrix, clearly has to be important. And unfortunately, not a simple drug target, but I think it is important for us to be able to understand that better. So most of the GWAS studies, we've got a SNP, and a SNP is related to a gene. It's close to a gene. It affects the expression of that gene. So in terms of expression, QTL analysis, etc. But what about the microRNAs? So Majid did this study a couple of years ago. It was published in Cardiovascular. in the ESC journal. So what he did a GWAS analysis for microRNAs. And what he did was to do, we had a set of microRNA-seq data, and that was actually done by Nestle in another study, and 1,000 G imputed genotypes, only 710 individuals. I mean, that's not many, but not many people have, microRNA-seq data in plasma. In fact, we were able to replicate in 2 data sets, Jubatus and Framingham, and these are much smaller data sets than we have. And so he was able to identify a GWAS significant signal, a mere QTL for 143 circulating microRNAs. And of course, it's limited to what's circulating in the plasma. And there's just a bunch of these, and they're all over the place. But I'll tell you, then he does some complicated bioinformatics analysis that only Majid can explain very well. I don't know if you'll kind of understand it. But this is summary-based Mendelian randomization. So what you do is to identify the associations between gene expression or microRNA expression and complex traits using summary data from GWAS, eQTL, mere QTL, et cetera. So you don't need all the individual genotypes. And then there is a fancy test called HI, and that distinguishes between pleiotrophy and linkage. And so he's able to look for the most functionally relevant genes at the GWAS loci. So then what he does is to integrate significant mere QTLs with previous omic findings by SMR analysis, trying to identify the microRNAs that mediates the effect of GWAS loci on blood phenotypes. So we're going from the gene to actually what might be linked. And of course, by now there are numerous GWAS data sets for a whole pile of cardiometabolic traits. I mean, we're mostly working on coronary disease and lipids, but it's out there for all sorts of things in terms of metabolism. And then a multi-SNP SMR analysis, that's 2-sample Mendelian randomization to investigate the causal associations using GSMR. So if you understood all that, you're way ahead. Okay. So I'm just going to focus on 2. So there's a bunch of mRNAs that are really interesting. These are the ones that we thought were most interesting. Mere 1908-5P, strongly associated with LDL, glycemic traits, fasting blood glucose, and A1C. 2 things there really interesting. We haven't followed up on these others, although some other people have. Mere 1993P associates with LDL, 10P5P associates with coronary disease. So what did we do from there? We started to look at the SNPs that came out, and basically these SNPs, the one that was sort of pulled out and the one that is in close equilibrium, 174.561C, strongly associate with the expression of mere 1908-5P. And that in turn, so this is the 2-sample MR, associates with total cholesterol, with LDL, and hemoglobin A1C, fasting glucose. So, and there is some secondary association with CAD. So that's that SNP and that mere sounded really kind of interesting. So we then followed up on that with some nice studies done by our former PhD student. And what she was able to show, so it appears that this microRNA is protected. So she was able to show that the minor allele of this SNP associated with increased expression of 1908 and lower level. So this is the protective effect of mu-1908-5P. And interesting that we expect the LDL cholesterol to be somewhat distinct from the effects in A1C. And she was able to show, linking the genetics to what's happening to the mu-1908, she was able to show that the protective G allele increased the expression of 1908-5P by, close to 2-fold, so really a pretty significant effect. She then went on to show, very interestingly, this mere is cholesterol responsive. So that's kind of unusual. And this was done either with cholesterol loading with methyl beta-cyclodexin or cholesterol depletion by treating cells with zuvastatin to decrease cholesterol production, and showed a nice reciprocal response there in terms of the change in total cholesterol. And that was limited to effects of 1908 3P rather than the 5P rather than the 3P. So how would this SNP possibly affect LDL? And we're working in LDL. I mean, I think the glycemic effects are also very interesting. And so the first thing she was able to show very interestingly, because we looked, does this mirror affect expression of the LDL receptor, any of the other genes that are related to cholesterol metabolism? And what happens at the, in terms of not only RNA expression, but more importantly, protein expression. And what she was able to show was that the mirror that's protective increased the ratio of the cleaved to full length LDL receptor. And well, how does this work? She was able, we knew from previous studies that DMT1 mediates LDL receptor cleavage, and that this mirror reduces, actually interesting, so it actually TGF beta 1 decreases TGF beta 1 expression. This is what leads to reduced BMP. And did some studies where adding exogenous TGF beta, she showed that that was able to ameliorate the 5P inhibition of LDL receptor cleavage. So a really nice mechanism that she worked out. And I think it's interesting. Is near 1908 5P a drug target? Probably not. I think it's very interesting that it actually links to TGF beta via BMP1, and TGF beta is a protein that we would like to follow up on, both in terms of the genetic relationship to TGFB1 expression and the mechanism by which it is modified. Okay. Well, that's just a little bit of a very quick coverage of functional genomics that we do in my lab, and it takes a long time to do it. But I think it is important. So I think the GWAS studies have really provided us, insights into biology. And as we'll say in a few minutes, the ability to create these polygenic risk scores that can basically improve our ability to assess risk a priori in a patient that's seen in the clinic. But it's really whole genome sequencing, done originally in unusual pedigrees, because that's how they discovered PCSK9, these French pedigrees with very high, very low LDL cholesterol, and what was the causative gene. More recently, whole exome sequencing in large populations. This is being done by Regeneron, a bunch of other groups. And so, if you have the resources, whole exome sequencing is not nearly as expensive as it used to be. We're able to identify rare variants or relatively uncommon variants that have a large effect size. And these are the ones that have led to discovery of novel drug targets. So, I mean, there are a host of them. And obviously, PCSK9 was one of the important ones. So this target drug discovery, I'm going to talk about ANGPTL3 today, but obviously PCSK9, one of the first genes that was shown to be strongly related to LDL cholesterol levels. ANGPTL3, really interesting. Other ANGPTL4 as well, although less has been done in that. ApoC3, so an important regulator of triglycerides. ApoA5, also a regulator of triglycerides. So these have a little bit of information from GWAS studies, but most of the new information here has been related to exome sequencing. What's really important, of course, if you're a drug manufacturer, is that genetic evidence markedly increases the likelihood that a new agent is going to be successful. So I think for many new agents, it would have been important to do the drug studies first. For example, CETP inhibitors, as you know, most of them have been a failure. Well, the GWAS studies did not suggest that CTP would be a particularly useful drug target. Neither did the studies in rare pedigrees, particularly in Japan with CTP deficiency. So I think you start with the genetics and then you think about new drugs. Okay, so whole exome sequencing. Been done in a lot of cohorts, but this is a paper that was published in the New England Journal 2017. And this is a collaboration between Geisinger and Regeneron. So Regeneron has lots of money and they are doing whole exome sequencing on almost every sample they can get their hands on. And we've sent them a number of samples over the course of the last few years. But this is a study where they had a bunch of different cohorts, but basically they did whole exome sequencing and found a number of things. But here we're focusing on ANGPTL3. And they're able to identify 13 different loci across, in the variants in ANGPTL3 in a total of 417 individuals. So, of course, some of these are small data sets. Like they only had 3 carriers, but across the board with meta-analysis, the individuals who had loss of function variants in ANGPTL3, not shown here, they had significantly lower levels of triglycerides, HDL, and LDL cholesterol, and definitely lower LDL prevalence, so that the odds ratio was approximately 0.7, so pretty good for just a single gene. So that made ANGPTL3 a very interesting drug target. So they, in the same work, they actually did mouse studies to sort of corroborate this, but they also did a study with a monoclonal antibody called Affymetrix, and various doses as they do in these things. But the 20 milligram per kilogram Affymetrix dose actually reduced triglycerides by 75%. So really very significant. Why is that important? Well, it's important as a clinician because we don't have good treatment for severe hypertriglyceridemia. There is a risk for atherosclerotic cardiovascular disease. Is it related to triglyceride? It's probably more related to the fact that triglyceride-rich lipoproteins carry cholesterol. So it's the non-HDL, the total cholesterol minus HDL, and the total cholesterol. It reflects the cholesterol in the triglyceride-rich particles. So it's a risk factor for atherosclerosis. But when triglycerides are greater than 10 and certainly greater than 20, there's a risk for pancreatitis. And that's the bad thing that nobody wants to get. People can go on, they can die, they can go on to chronic pancreatitis. So not a good thing. This actually is a patient that I saw not long ago. Actually, a young guy comes in and he's got these lesions all over. And his family doctor thought it was some kind of acne and sent him to a dermatologist who said they're not acne, they're just fat. So anyway, turned out he had sky-high triglycerides. He hadn't developed pancreatitis, but well, he knew onset diabetes, didn't know that he had diabetes. And of course, hyperglycemia, the liver, great substrate for triglyceride synthesis is glucose, other things such as alcohol. So we were actually able to get his triglycerides down without too much trouble. But for a lot of people, we simply can't get triglycerides low enough. You can use fibrates, you get maybe a 35% reduction in triglycerides. Obviously, controlling diabetes is important, high-dose statins. But I think really interesting here, this monoclonal antibody reduced triglycerides by 75%. So it could be a really useful additional treatment for severe hypertriglyceridemia, and particularly those patients who are at risk for pancreatitis. So not yet available in Canada because in Canada, it takes a long time to get any new drug here. In a separate study, and this is really interesting, and this is about 3 years later, this is the same group with the monoclonal antibody Affymetrix. They did a study in homozygous FH. So people with homozygous FH are just dripping central all over the place. Without treatments, they tend to develop coronary disease or carotid disease, a pretty aortic stenosis in their teens, sometimes earlier. I have 2 patients with homozygous FH, and the only way to treat them, because they don't have LDL receptors, so if they really have 2 null alleles, they have no LDL receptors. So statins, all the drugs we have work by upregulating LDL receptors, gene expression, or increasing the functionality of LDL receptors by inhibiting PCSK9. But what if you have no LDL receptors? So there's not much you can do except apheresis. So these 2 young women are having plasmapheresis every 2 weeks. Plasmapheresis, which is not a pleasant thing to have, and a problem now because we're running out of albumin, but plasmapheresis because we don't have funding for LDL apheresis, another way to just remove the LDL and not remove all of the lipoproteins. Nonetheless, plasmapheresis works, and these xanthomas do start to melt away. But of course, we never get LDL down to where it should be. We get it from 18 down to about 7, but that's not very good. Okay, so what was really interesting they showed was that they took these people, these young individuals with homozygous FH, treated them with evangelization, and there's this quite amazing, on average, about a 50% decrease in LDL. So this means we've got a new therapy for homozygous FH. It probably won't erase the need to use plasmapheresis, but certainly it is a new therapy that at least could reduce the frequency of apheresis and hopefully get these people to an LDL where they're not going to continue to have cardiac events. So we don't know how it works. I mean, that's what's really fascinating from the lipid point of view. So ANGPTL3 acts to inhibit lipoprotein lipase and endothelial lipase. So with evangelization, you're increasing the lipolysis of the LDL via lipoprotein lipase, also increasing endothelial lipase, which in turn alters the LDL composition. And it's thought, well, maybe this enhances the clearance of these particles more quickly so that they're never converted to LDL. There's conflicting data in terms of the effects of ANGPTL3 inhibition on the LDL secretion, although there may be an effect there. So we've got this really interesting treatment. We don't know how it works exactly, but certainly it does work. And I think very exciting. So right now, these two people I have with homozygous FH, the company has agreed to provide evangelization free of charge. We still get Health Canada to approve it. So it's just crazy politics we have in the government in Canada. The other point to make, I mean, monoclonal antibodies, are effective, they're safe, and of course, we're using them for PCSK9 for a long time. But of course, they're expensive to make. You're making these big vats of monoclonal antibodies. So another approach to targeting the genes that are of interest in terms of a particular phenotype is the use of antisense nucleotides. So this is being done right now for PCSK9. So there is a drug called inclisiran, Leqvio, that was developed by Ionis and very effectively lowers LDL by about 50% as compared to 60% for monoclonals, but obviously should be a lot less expensive, injected every 6 months and simpler. But once again, well, Leqvio is just now available in Canada, but it's not approved by CADIS, so it's not covered by ODB, by a little bit of progress there. So the antisense, they're basically, those who have to understand it, small interfering double-stranded RNA. And so basically what they are doing is binding to the RNA, preventing in the end protein translation. So it's really, using the natural process of RNAi. The way it works, these nucleotides are modified so that they have low immunogenicity and they're durable, so they're not immediately destroyed. So for the most part, they'll last for a month, or with inclisiran for 6 months. Really important, they're distributed to the liver via GalNAc conjugation. So that's important. So if you think, for example, PCSK9 is important in some other tissue, the antisense is not going to get there. For ANGPTL3, I'm not sure if it matters, but anyway, it's going to the liver. It's only acting in the liver and nowhere else. So I think this is really not entirely new, but it's a form of therapy I think we're going to see more and more, not just for PCSK9 and TL3, but also ApoC3. There's been a bit more problem developing the ApoA3 antisense, but also it's turning out to be quite effective. Okay, no one wants to hear about Mendelian randomization anymore. Do you? It's very simple. It's very simple. It's very simple. I mean, so we talked about drug discovery and making sure that the drug target is the appropriate target, but basically it's very simple. So, do GWAS studies, and you've got these genetic variants that associate with the biomarker, and it could be anything. And the biomarker may associate with disease, but is that a genetic relationship, or it's due to something else that might affect that biomarker, and the biomarker per se has nothing to do with the disease? So it's kind of a simple paradigm. So if we take something like LDL. So we have these genetic variants near the LDL receptor, PCSK9, ApoE, SORT1. They associate with LDL, but they independently associate with coronary disease. So really, this is likely to be causal. Well, we know LDL is causal, but we find the same thing in terms of genetic variants associated with lipoprotein A, adiposity, A1C, interleukin 6, which obviously gets into the importance of inflammation and immunity. But it's not true for CRP or LPPLA2. And this is important because there are two drugs developed to inhibit LPPLA2. They underwent huge, big randomized controlled trials that were negative. And I think if those investigators had looked at the genetic data, the Mendelian randomization data, they probably never would have done those studies. And of course, what we learned through that is that we do a study giving a statin for 5 years, and we do reduce the risk of coronary disease, 30% in the early studies, 50% now, but it's not the same as having a low LDL all of your life. So the genetic variants predict risk much better than simply lowering LDL over a short period of time. So we always talk about life years of LDL. It's like pack years of smoking. So when I see a 30-year-old with familial hypercholesterolemia, we're trying to get LDL very low to compensate for the fact that that person has been untreated for 30 years and has been running around with an LDL well above the 99th centile. And finally, this is what we can use the data from the GWAS studies for, and that is assessing polygenic risk. So this has been developed over the course of the last many years. Early on, we only had a few SNPs, so we tried to do a genetic risk score with a few significant SNPs, and then we started using more and more as it came along. But of course, you can include genomide SNPs with don't worry about the PR, just put everything in there. And so you do a meta-genomide association score. So this one published in JACC by Ruddy about 4 years ago included almost 2 million SNPs. And really it's going to give you more information than those using just a few SNPs. But going back, because I always have to talk about that later, this is Sunny Danota, who worked with Alex and the group a few years ago. And he actually viewed all of these angiograms as nice data. And basically what he was able to show was that for individuals who had no risk variance, the disease severity, most of them had no disease. As you go up to, having more like people who are heterozygous versus homozygous, the more risk alleles you have, you're more likely to treat lots of disease. So obviously there is a close relationship there. And that's 9p21 because it has a pretty significant effect for a common allele. But other studies have done this. Was work from Sig Kathirason's group a few years ago, and they took 3 studies. So that was the ERIC study, the Women's Genomic Health Study, and the Malmo Diet and Heart Study. And they only used 50 SNPs because that was what we had in those days. And they're able to show in all of these studies that people in the top tertile, wherever the study, had about a 2-fold increase in risk. So it really gives us information, and this is after correction for known risk factors. So really gives us a lot of useful information. And when they then went on and did a polygenic risk score, once again going genome-wide, they were able to show that individuals really at the extreme end of the polygenic risk score, the top 1%, had a 5-fold increased CAD risk. So, I mean, that's maybe a small number of people, but very significant. And I think really showing us that this can be useful. There's always arguments, well, how much is this going to add to what we do already in the clinic? But, these polygenic risk scores were corrected for known risk factors. I think very important. And of course, because I want to tell our patients that genetic risk is important, but it's not the end of the world. So this is a study by Hindi a couple of years ago. And once again, using tertiles of a genetic risk score. So, and according to smoking. So low risk non-smoker, that was the lowest level. And even if you're a non-smoker, risk goes up as into a higher tertile or polygenic risk score. But obviously that risk is much more significant for individuals who smoke. So I suppose one message may be if you tell people they have a high genetic risk score, then maybe they can modify these other things that are important, such as cigarette smoking. And also getting back to the study I showed you previously from Pearson and Kira. Once again, these three studies, Eric Rome Health Study and the Malmo Diet and Heart Study, you know, if you look at genetic risk from low, intermediate to high, there is an increase in risk. But for individuals who have a favorable lifestyle, here in blue, versus an unfavorable lifestyle, there is a very significant difference. So basically, I think you can tell people if they have high genetic risk, you know, it's really much more important to modify lifestyle to at least get them down to a more modicum level of risk. So does that work? I think it works for some people, not everyone. So it's unfortunate we don't do this because, you know, we could actually do it in-house. So how do you create a genetic risk score? Or you just run on the chip, and the chips, if you consider the cost of the chip and then the costs of bioinformatic analysis, the kind of stuff that Majid does, I mean, it's less than $100 to get, you know, enough genetic markers to create a polygenic risk score for anything that you like. So I think eventually this should be routine. I think perhaps it isn't because of reticence regarding genetic testing, reticence regarding genetic results. So even though we're looking here at common genetic variants rather than something important like carrying the ApoE4 allele or having a mutation in a gene that's going to affect morbidity in a significant fashion, these are common variants. So I think it's something that could be done, but we could do this in-house. And what's also neat about it is all the genome-wide markers, but you can also in this chip, you can insert clinically relevant variants from ClinVar so that you can look at risk variants associated with, you know, hypertrophic cardiomyopathy, a number of things that really are important that we'd like to know about. And also pharmacogenomics, because there's a difference in metabolism of a whole pile of drugs. And of course, the one we know best is clopidogrel, but there are a number of others such as warfarin. It'd be nice to have that so you can customize them. Anyway, that's the future. I wish we could do it, but we're not doing it yet. These are the people in my lab who've done all the work, and Alex St. Sebastian particularly, and these are the people around the world that we've collaborated with. Thank you.
All right. Thank you, Ruth. That was an excellent presentation. Now, if we have anyone in the audience who would like to ask a question, you can raise your hand. And do we have anyone present? Okay, Al.
Thanks, Ruth. Is this on?
Yeah.
So I was wondering about pleiotropy. I mean, so you presented, you know, a lot of genome-wide association studies, and you also talked to us about Mendelian randomization if you control for a certain trait.
Yeah.
But I mean, these genetic variants affect so many. It's not just one gene, one trait. It's many traits often.
I know.
And so, yeah, is part of our problem that so we have 234 so far genome-wide significant SNPs for coronary artery disease, most of them with fairly small effect sizes, but maybe we haven't delved into those sufficiently to get a better feel on mechanism. Because I remember several years ago, I came up with a genetic variant that looked promising in these genome-wide association studies in the SPG7 gene.
Right.
And then it turned out, well, it also affected blood glucose, and it also affected the response to anti-cancer drugs in prostate cancer patients. So it was having all kinds of effects. And maybe the reason it never panned out to a genome-wide significant level was because of all these other effects. And I'm just wondering, Isn't it our problem that we just don't know enough?
Well, we certainly don't know enough. And, you know, in terms of causality, maybe it's really simple if you, well, as Aaron was saying at the start, maybe PCSK9 does a bunch of other things, but, you know, it's kind of simple if, well, the LDL receptor gene, that's LDL. Yeah, you know, there's always some pleiotrope, and even we sort of argue that perhaps there isn't, but yeah, that's certainly an issue. And of course, as you've alluded to, the more complex issue is the fact that, you know, these genes are affecting many processes. So different in place and time, developmental effects, cell-specific effects. And, you know, we may, you know, we do EQTL studies, you know, in certain tissues, but we're only starting to get EQTL data at the cellular level. And even at the cellular level, it's kind of static, fixed in time. So there's a lot of missing information. So, I mean, I think it's why I say generally the GWAS studies have, you know, really given new attention to things like, you know, the vessel wall, but, you know, mechanisms are still unclear, and probably unclear because they're very complicated. And maybe maybe the best use of GWAS data is the polygenic risk score, because for whatever reason, if it tells you that, that's useful. And I think the whole exome sequencing is useful because it's very specific. Remember when we started to do the GWAS studies, the argument was that after years of sequencing genes, Sanger sequencing, you got nowhere at all. But now that we can do the sequencing in, you know, huge populations, suddenly it's becoming, you know, much more useful. But yeah, there's still a lot to do for sure.
Thanks.
Okay. From Dr. Beanlands, he says, great work and leadership, Ruth. You've kept us at the cutting edge in this field. I think he's articulating well what a lot of us feel. And then, is there a role for genetic testing to define targets for LDL reduction, or when to consider inhibition of PCSK9 other than for FH?
Yeah, so PCSK9 is a great drug target. I think it's a really good drug target because it, you know, it's pretty clean. At least the monoclonal antibodies, you know, really a nice effect. And of course, you know, one problem we have today is that for patients who do not have a good private plan, because the cost of PCSK9 inhibitors are $3,000 to $6,000 a year, ODB for individuals over the age of 65 or on ODSP or something, the coverage for PCSK9 inhibitors only for patients that's bona fide FH. So they have to be those unusual people who start off with no development of 10. And the individuals who really need it are patients with coronary disease. And even you have a patient with CAD and their LDL is 1.8, but if they have had 3-vessel disease that's developed over many years, if they have saphenous vein grafts, if they have multi-vessel disease, carotid peripheral arterial disease, you know, that LDL 1.8 isn't nearly low enough. So we always try to get it better. So one would hope, and maybe when inclisiran becomes used, I think Novartis is willing to really move on the price so that individual payers may not pay that much for inclisiran or Leqvio as compared to the monoclonals. But yeah, I think many of these people need it. The other question is who needs it? Because, you know, I see a lot of patients in my clinic, secondary prevention, so it's very clear you want to get everything low. But I will see patients who are referred because a very high level of LDL cholesterol, maybe some other risk factors, and they're already on a statin. Do they need a PCSK9? So some of those patients all actually do a CT angiogram. And it's always surprising because you do have patients with, you know, very high LDLs. They have clean coronary arteries. So, you know, you kind of stop there. If they're 50 years of age, you continue to obviously treat their cholesterol. If they're 80, then you can probably forget about them. So, you know, obviously having some information on the disease is, I think, really important in that decision. And it's important in the primary prevention, so-called cohort.
And maybe I'll just ask you, what do you think of the reports that PCSK9 inhibitors can lower Lp(a)? Do you think there's any?
They do lower Lpa, yeah. So PCSK9 inhibitors, the reduction in Lpa 25 to 30%, not great. And of course, Novartis has a antisense RNA that is being tested. We were part of the phase 2 study. So the Novartis antisense against Lpa reduces LPA levels by up to 85%. So not available for prescription yet because they are doing a study. So the study is in individuals with established CAD, a lipoprotein A level greater than 70 milligrams per deciliter, which is about 130 nanomoles per liter, which the labs are now reporting. If that study is positive, then I think we will have that treatment for, because we do see these patients extraordinarily high levels of LPA and certainly an important causative risk factor for their disease. So more antisense coming along.
Yeah, that's great. Really exciting. All right. If there are no further questions, I'll ask everyone to join me in thanking Dr. McPherson for an amazing lecture and look forward to next week's lecture.
Thank you.